Blood moves quietly inside you, so familiar that you rarely stop to wonder what it truly is. Yet if you could step inside your own veins and drift with the current, you might feel as though you were entering an ancient river, one that began long before humans, long before mammals, long before anything with a heartbeat existed. It is a river shaped by catastrophe, chance, and relentless struggle. Every pulse you feel today is powered by victories won by creatures you will never know, ancestors who lived and died hundreds of millions of years before your story even began. And the blood flowing through you remembers them, in ways you may never have imagined. When we picture blood, we usually think of the obvious, a bright red liquid that carries oxygen across the body. But that description barely scratches the surface. Blood is not merely a transport system. It is a living archive. It is a survival engine. It is a restless, constantly moving ecosystem made of countless cells and molecules, some ancient, some newly formed, all of them playing out dramas that echo the very earliest struggles of life on Earth. Your blood holds bacterial ancestors, viral fragments, proteins older than the first fish, and minerals forged in dead stars. It is a fusion of cosmic history, evolutionary refinement, and personal experience, all circulating beneath your skin. Imagine following a drop of your own blood as it leaves your heart. It rushes out with astonishing speed, propelled by a force that began evolving more than 500 million years ago. The moment it enters the arteries, it's surrounded by red blood cells, tiny concave discs packed with a molecule capable of performing one of the most remarkable tricks in nature. Hemoglobin, with its 4 iron atoms, binds oxygen with precision so delicate that it can cling tightly in one environment and release instantly in another. This chemical choreography is not an accident, it is the outcome of countless evolutionary experiments, most of which failed. You are the beneficiary of the version that worked. But your blood is far more than red cells. It carries immune warriors, white blood cells, that inherited instincts from early lifeforms that once patrolled ancient seas. Some of your immune cells behave like solitary hunters, stalking invaders with eerie patience. Others act like coordinated armies, communicating through chemical signals that function like alarms and tactical orders. Their strategies come from an evolutionary arms race so old that the creatures involved have no names, no fossils, and no stories outside the silent memory preserved in your bloodstream. Floating among them are platelets, fragments of cells that rush to seal wounds with astonishing speed. They act with no consciousness and no command, yet they behave with precision any engineer would admire. Their ability to recognize danger, assemble rapidly, and create barriers is the reason your ancestors survived predators, accidents, childbirth, and wars. Without them, human history would be impossible. Even the liquid part of blood, the plasma, tells an ancient story. Its composition closely mirrors the chemistry of Earth's early oceans. In a way, every heartbeat sends a small piece of the sea through your body, a reminder that life began in water and carried that heritage inward when it crawled onto land. Sodium, chloride, potassium, calcium, these ions drift through you like echoes of the primordial world. You are, quite literally, an ocean folded inward. As your blood continues its journey, it brings food to your cells, heat to your extremities, and chemical messages that orchestrate everything from your mood to your metabolism. These signals shape your feelings in ways you seldom notice. A surge of adrenaline during fear, a rise in endorphins during wonder, a flood of cortisol during stress, all of these emotional currents flow directly through the same river that once kept your distant ancestors alive when predators stalked them in the dark. You do not simply feel emotions, your blood participates in them. Yet blood is also deeply personal. It carries genetic instructions from your parents, immune memories from every illness you've ever survived, and subtle signatures of the environments you've lived in. Every sleepless night, every moment of joy, every wound, every infection, every season of your life, trace evidence of all of it circulates in your veins. Blood is not just a biological substance, it is the ongoing story of you, written in proteins, minerals, antibodies, and cells. Still, the most astonishing truth is that none of this was inevitable. The river inside you could have evolved differently. It could have been thicker, thinner, colorless, or based on copper instead of iron. Countless species took different paths. Spiders and octopuses use blue blood. Some insects circulate simple fluids without red cells at all. Many paths were available, but the one leading to human blood was shaped by choices made by ancient fish, amphibians, reptiles, and mammals long before humans existed. Their triumphs and failures shaped the oxygen levels in the environment, the demands of movement, the pressure of gravity, and the arms race with parasites. Your blood still carries the results of those battles. Pause for a moment and feel your heartbeat, not as a mechanical sound but as a reminder that what moves inside you is older than mountains, older than continents, older than the moonlit skies your ancestors once looked up at in wonder. Your blood is not just yours. It is the sum of billions of years of survival, adaptation, and transformation. It is a river carved through time, carrying every secret that life has ever learned about how to endure. And all of it is flowing inside you at this very moment, quietly, tirelessly, telling a story far deeper than you ever realized. Long before blood existed, before the first heart pulsed, before any creature gasped for breath, life began as something so small, so delicate, that it could barely be seen even with a modern microscope. These early beings floated through an ancient ocean that was nothing like the one we know today. The water was thick with minerals, chemicals, and drifting particles left behind by volcanic eruptions and meteor impacts. Sunlight struggled to reach the depths. Lightning storms cracked across the sky, sending energy into the sea. And in that restless, chaotic world, the first primitive cells emerged, each one holding the faintest spark of what would someday become the story of every living creature on Earth. These earliest forms of life had no blood, no veins, no circulatory systems at all. They survived by relying on simple diffusion, the slow, passive movement of molecules across their membranes. Food, gases, and waste drifted in and out of them without any pump or coordination, carried only by the gentle stirrings of the water around them. Nothing rushed. Nothing flowed. Life was slow, quiet, and utterly dependent on the rhythms of the ocean. In a way, these organisms lived inside the world's first bloodstream, the sea itself, a vast, swirling medium that delivered everything they needed and swept away everything they did not. For millions of years, this primitive existence was enough. Life grew, diversified, and experimented with new shapes and strategies. But as organisms became larger and more complex, they faced a new challenge. Diffusion alone could no longer sustain them. Their bodies had too many cells, too many layers, too much internal space for nutrients to reach every corner without help. Evolution does not provide warnings, only consequences. And as complexity increased, so did the pressure to find a new way to transport life's essentials. Imagine a simple worm-like creature emerging on the ocean floor, its body no longer just a tiny sphere but a long, flexible tube. Food couldn't simply drift to every cell anymore, oxygen couldn't seep inward on its own. These organisms needed an internal ocean, a small, enclosed environment where water and nutrients could circulate. And so, slowly, evolution began to carve channels inside their bodies. Fluids moved from one end to the other, pushed not by a heart, but by gentle waves of muscle contraction. It was crude, slow, and imperfect. But it worked. These were the first steps toward internal circulation. As the oceans changed and life diversified, these early creatures faced new predators, new habitats, and new environmental pressures. In response, internal fluids became more specialized. Cells suspended in the fluid began performing specific tasks, carrying nutrients, removing waste, transporting signals. But these cells were still nowhere near what we recognize today as blood cells. They were merely pieces of a puzzle evolution had only just begun to assemble. The Earth itself was transforming too. Volcanoes poured minerals into the sea, meteor strikes brought new chemical ingredients, and the atmosphere slowly shifted. With each change, life had to adapt. Some organisms developed thicker protective layers, others evolved new ways to move, hunt, or escape. These behaviors demanded more energy, faster reactions, and better internal transport. The ocean outside was no longer enough. A new internal sea was required, self-contained, regulated, and capable of delivering life's essentials on demand. Picture the ancient world during this period. Supercontinents drifted across the planet, splitting and colliding. Shallow seas teemed with strange creatures, trilobites with armored bodies, jelly-like animals drifting silently, small worm-like beings burrowing into soft sediment. Each one carried a living fluid within it, but no two species carried the exact same kind. Some had clear fluids, others milky or yellowish. Some had cells that clumped together, others had none at all. These were evolutionary experiments, each testing a different strategy for survival. But the ocean outside still played a guiding role. Its chemical composition shaped the internal fluids of these early animals. Sodium, potassium, and chloride, ions essential to modern blood, originated from the sea. It is not a coincidence that human plasma retains a balance of salt strikingly similar to ancient seawater. Life did not abandon the ocean, it carried a piece of it inward, making it portable. The need for movement increased as predators grew faster and prey became more evasive. Creatures developed muscles, and muscles demanded oxygen. But oxygen did not dissolve well in basic body fluid. Evolution needed something better, something capable of grabbing oxygen, holding it tightly, and delivering it wherever it was required. And so began the long, slow development of primitive oxygen-binding molecules. These molecules were the great-grandparents of hemoglobin, the red pigment that defines our blood today. But in these early days, nothing was red. There was no color, no iron-based protein, no crimson tide flowing through arteries. Just simple molecules learning to interact with oxygen, solving one tiny problem at a time. As life continued to grow more complex, the internal fluids of these creatures began to resemble the earliest versions of blood, not in appearance, but in purpose. Transport. Nutrition. Defense. Communication. These roles emerged gradually, like instruments entering an orchestra one at a time. No single mutation created blood. It was a long chain of small solutions, each building upon the last. Think of the millions of years that passed during this stage. Entire species flourished and disappeared without ever developing a true circulatory system. Others took steps forward only to reach evolutionary dead ends. Yet every experiment, every failure, contributed something to the story. Evolution does not plan, but it accumulates, remembers, and repurposes. And slowly, the internal seas of early creatures became more specialized, more efficient, more coordinated. Somewhere in that ancient world, no one knows exactly where or when, the foundations of real blood began to form. A fluid that circulated. A set of cells that performed specific tasks. A pump that kept everything moving. Nothing like the blood found in humans yet, but the blueprint was emerging. The earliest hints of the vital system that would one day sustain creatures capable of thought, emotion, imagination, and reflection. This age before blood is easy to overlook, but without it, nothing that came afterward would exist. The oceans nurtured life, shaped it, and then inspired the internal systems that allowed animals to explore new worlds. Before blood could become the river that carries oxygen through human veins, it had to be a drifting molecule in the sea, a simple fluid inside a worm, a primitive system inside a creature that left no fossils behind. What came next, the true invention of blood, would transform everything. But its story begins here, in the quiet, ancient world where life first learned to flow. For billions of years, life drifted peacefully in Earth's early oceans, surviving on simple chemistry and steady routines. But then, something extraordinary happened. something so dramatic that it nearly erased life entirely. It was a transformation that arrived quietly, through tiny organisms no larger than dust motes, yet it reshaped the entire planet. This was the Great Oxygenation Event, a turning point when oxygen, now essential to us, first appeared in the atmosphere as a toxic, deadly gas. To understand your blood today, you have to understand this ancient catastrophe, because your bloodstream is, in many ways, a direct response to a world that suddenly turned dangerous. Before oxygen flooded the atmosphere, early Earth was a place of chemical shadows. Volcanoes poured methane and sulfur into the air. The oceans were rich with iron and minerals but poor in anything resembling breathable gas. Life did not need oxygen. In fact, oxygen was so reactive and corrosive that it would have destroyed most early cells instantly. The first living organisms were anaerobic. They thrived without oxygen and considered it, quite literally, a poison. For them, the world was stable, predictable, and safe. But deep in certain shallow waters, something new was stirring. Cyanobacteria, tiny blue-green microbes, began performing photosynthesis, using sunlight to split water molecules and release oxygen as a byproduct. At first, the oxygen remained trapped in the oceans, reacting with dissolved iron and forming rust that settled to the seafloor in colossal layers. These banded iron formations, still visible on Earth today, are ancient scars left by that first encounter between life and oxygen. Eventually, the oceans could hold no more. Oxygen began to seep into the atmosphere, molecule by molecule. What followed was one of the most devastating extinction events in Earth's history. Species that had thrived for millions of years found themselves engulfed by a toxin they could not tolerate. Oxygen tore apart their molecular structures, damaged their proteins, and destabilized their ways of life. To them, the changing world was burning from the inside out. But evolution is not just destruction, it is opportunity. Some organisms, by luck or by ancient genetic quirk, possessed molecules that could neutralize oxygen's corrosive effects. These molecules became early antioxidants, the ancestors of systems your cells now use every second. Organisms that mastered these defenses survived. Those that went further, using oxygen to generate energy, came to dominate the planet. This was the moment the story of blood began to shift. Oxygen was no longer merely a toxin, it was a power source. Using oxygen, cells could extract far more energy from food than ever before. But there was a problem. Oxygen was scarce, unevenly distributed, and difficult to move through tissues. Diffusion alone was too slow. Cells deep within larger organisms could not access enough oxygen to survive. The New World demanded a solution, a way to transport oxygen quickly and safely. Imagine early multicellular life struggling through this transition. Their outer cells touched the oxygen-rich environment, but their inner cells suffocated. They needed an internal logistics system, something that could grab oxygen from the environment, carry it deep inside the body, and release it where it was needed. This challenge pushed evolution toward an innovation that would ultimately define complex life, specialized molecules that could bind oxygen. At first, these molecules were primitive, simple pigments capable of holding small amounts of oxygen. In some early species, these pigments were stored in the body fluid itself, tinting it faintly. Later, they were packed into dedicated cells, creating more efficient transport. And slowly, with each evolutionary refinement, the framework for hemoglobin began to emerge. But oxygen brought another danger, the risk of runaway chemical reactions. Too much oxygen could damage cells as easily as too little. Organisms needed a way to control its delivery with exquisite precision. They developed structures that could change shape depending on the amount of oxygen around them, binding it in one environment and releasing it in another. These early systems laid the groundwork for the sophisticated oxygen handling mechanisms inside your blood today. Meanwhile, the atmosphere continued to transform. Oxygen levels rose, then fluctuated wildly for millions of years. These swings created selective pressure unlike anything life had faced before. Species that adapted gained new abilities: movement, predation, speed, endurance. Species that could not adapt disappeared entirely. Over time, oxygen became the defining force behind biological energy systems, shaping everything from metabolism to behavior. Picture the world during this transition. The skies grew clearer as oxygen altered the chemistry of the atmosphere. New minerals formed, many of which exist only because oxygen interacted with Earth's crust. Lightning storms behaved differently. Perhaps most importantly, the ozone layer slowly began to form, shielding the surface from harsh ultraviolet radiation and making life on land possible. But even with all these environmental transformations, the most profound changes were happening inside living creatures. They were developing internal fluids optimized for oxygen transport. Iron, abundant in the oceans, became essential because it could bind oxygen more effectively than almost any other element. The choice of iron, made billions of years ago, still determines the color of your blood today. The organisms that gained mastery over oxygen thrived. They grew larger, moved faster, and explored new ecological niches. They became ancestors of animals that breathe, move, hunt, and eventually think. Without the challenge oxygen imposed, there would be no need for the circulatory systems we rely on today, no need for hemoglobin, no need for the intricate red river inside you. It is strange to realize that the very substance you depend on for life began as a poison. And yet, this paradox sits at the heart of biological evolution. What was once destructive became essential. What was once feared became embraced. Life did not run from oxygen, it learned to harness it. Your blood is the final product of that ancient negotiation. Every breath you take, every beat of your heart, every cell that depends on oxygen owes its existence to organisms that survived one of the greatest environmental upheavals in history. The Great Oxygenation Event did not just change the atmosphere, it reshaped the destiny of life. And in doing so, it whispered the first hints of the story written in your veins. Life after the rise of oxygen entered a new and uncertain phase. The smallest organisms had adapted to this strange, reactive element, learning to harness its power while shielding themselves from its dangers. But now, evolution pushed life toward greater complexity, and complexity demanded a more sophisticated way to move oxygen, nutrients, and waste throughout the body. This was the era when the world saw the first attempts to create something resembling blood, not the red, iron-rich fluid humans carry, but primitive, experimental versions that lived inside creatures whose names and shapes are lost to time. These early fluids were the ancestors of modern circulation, quietly shaping the destinies of every animal that would ever walk, swim, or fly. Imagine the ancient oceans as vast laboratories. Within them lived soft-bodied creatures, worms, jelly-like organisms, early multicellular beings, that relied on simple internal fluids to keep themselves alive. These fluids were not blood. They had no color, no specialized cells, no ability to bind oxygen in any meaningful way. They were more like watered-down versions of the ocean itself, trapped inside delicate bodies. Nutrients drifted through them slowly, following random paths, reaching cells only by chance and proximity. Life at this stage moved in slow motion, constrained by the limits of diffusion and the absence of any organized internal transport. But as organisms became larger and more active, randomness was no longer enough. The first innovations were simple: channels or cavities running through the body, allowing fluid to move more predictably. Muscles squeezed these channels in rhythmic waves, pushing internal fluid from one region to another. It was not yet a heartbeat, but it was a beginning, a hint of circulation. The creatures that used these systems could grow larger, move further, and survive in more diverse environments than their simpler relatives. Inside these early bodies, the internal fluids began to change. They started to carry more than dissolved nutrients. Bits of cellular material floated within them, particles that helped repair tissue damage, molecules that transported minerals, and tiny, shapeless cells that performed the earliest versions of immune defense. Life was improvising, taking pieces of old systems and reassigning them to new roles. What began as a simple internal ocean was slowly becoming an organized medium, capable of supporting organs, movement, and faster metabolism. One of the key evolutionary experiments during this time was the development of oxygen-binding pigments. These pigments were crude precursors to modern molecules like hemoglobin and hemocyanin. They were not specialized yet, nor particularly efficient, but they provided a crucial advantage. In low-oxygen environments, they could cling to oxygen molecules long enough to deliver them deeper into the body. The color of these early pigments varied. Some were yellowish, some green, some faintly red. None had the vivid crimson humans associate with blood today. But their purpose was clear: to bridge the growing gap between oxygen supply and metabolic demand. As these pigments improved, organisms could become more mobile. Movement required energy, energy required oxygen, and oxygen required transport. Step by step, the internal fluids of ancient creatures transformed from passive mediums into active partners in survival. Fluid that once merely filled space now carried chemical messages, regulated internal balance, delivered oxygen, and supported cellular communication. Complexity beget more complexity. Then came the emergence of hemolymph, the blood-like fluid still found in insects and many invertebrates today. Hemolymph represents one of evolution's earliest fully realized models for internal transport. It is not confined within vessels the way vertebrate blood is; instead, it bathes organs directly, flowing freely through open cavities. Though primitive compared to closed circulatory systems, hemolymph proved incredibly versatile. It could carry pigments, nutrients, hormones, and even clot to seal wounds. For many species, it was more than enough to thrive. Within this expanding diversity of life, evolution continued its relentless experimentation. Some creatures experimented with copper-based oxygen carriers, producing blue blood. Others refined iron-based systems that produced pale or yellow fluids. Still others abandoned oxygen-binding molecules entirely, relying on thin bodies or slow lifestyles to avoid the need for specialized transport. Every strategy was tested against the demands of survival. Yet the most transformative innovations were taking shape in early chordates, the distant ancestors of fish and, ultimately, humans. In these creatures, circulation became more efficient. Fluids moved through tubes rather than open cavities. Muscular pumps grew more coordinated. Oxygen-binding pigments improved dramatically, setting the stage for the emergence of true red blood cells. But at this stage of the story, long before hearts, long before complex vessels, these were still experiments. Proto-blood was a mixture of ancestry and possibility, an evolving fluid learning new skills as life pushed toward new horizons. Its purpose was not yet refined, but its impact was immense. Without these early steps, the crimson river that flows through human veins could never have existed. The invention of circulation marked one of the most decisive steps in the story of life, a leap that allowed creatures to grow larger, move faster, and inhabit new environments. Before this innovation, internal fluids drifted slowly through tissues, relying on diffusion and chance to reach every cell. This method was sufficient for simple organisms, but as bodies became more complex, more layered, and more active, diffusion could no longer meet the growing demand. Cells starved deep within tissues, and without a solution, multicellular life would have remained constrained to small, slow forms, forever bound to the surface of the oceans. Evolution, however, had begun to experiment with something radically new: movement powered from within, the first primitive pump. Imagine an early worm-like creature crawling across the ocean floor. Its body is long, flexible, and segmented, with an internal cavity filled with a watery fluid. Muscle contractions along the body walls begin to push this fluid forward, then backward, in a rhythmic pattern. Each squeeze moves oxygen, nutrients, and signaling molecules closer to the cells that need them. This simple motion is the ancestor of a heartbeat. It is not yet a heart as we recognize it, nor is it a closed system with vessels, but it introduces the first element of controlled circulation. The fluid is no longer entirely dependent on the outside world or passive diffusion, it is now guided by the organism itself. This primitive circulation comes with immediate advantages. By propelling internal fluid, the organism can grow longer and more active. It can explore the environment for food and escape predators more effectively. Internal cavities filled with moving fluid also allow waste products to be carried away, reducing toxicity and enabling higher metabolic rates. Life at this stage becomes faster, more efficient, and more ambitious. The rhythm of these muscular contractions is the first echo of the pulse that will one day define hearts. As organisms evolved, these internal movements became more coordinated. Some segments of the body began contracting in sequence, creating waves that could push fluid more consistently in one direction. Even without a dedicated pump, these sequences allowed for rudimentary circulation that far outperformed random diffusion. The advantage is immediate. Creatures with coordinated fluid movement can sustain more active tissues, respond quickly to environmental changes, and survive in habitats that would have been inhospitable to slower forms. Circulation is no longer optional, it becomes a requirement for survival in an increasingly competitive world. The next evolutionary leap occurs when specialized structures begin to take shape. Small chambers within the body form, enhancing the efficiency of fluid movement. Early valves, simple flaps of tissue, prevent backflow, ensuring that the internal current moves in the intended direction. These first proto-hearts are not confined, fully formed organs, but they represent the first design principle of what will one day be the human heart, a muscular pump driving fluid through a coordinated system. The pulse that begins in a simple worm-like ancestor echoes through millions of generations, becoming more refined with each mutation and adaptation. Even at this stage, evolution experiments with multiple approaches. Some organisms develop open systems where fluid flows freely around tissues, as in modern invertebrates. Others begin enclosing their channels, an early attempt at a closed circulatory system that will one day dominate vertebrate life. Each system has trade-offs. Open systems are less efficient at oxygen transport but simpler and more robust. Closed systems require more energy and structural complexity but allow for greater specialization, higher metabolism, and increased activity. Both strategies leave traces that persist in the diversity of life today. The invention of circulation also changes the relationship between size and activity. Small creatures could survive without moving fluids, but larger organisms now find that internal movement is essential. Muscles can work more powerfully because oxygen and nutrients reach them reliably. Sensory organs can develop because they receive the resources they need to function. Even the earliest nervous systems benefit, as neurons demand consistent energy to transmit signals. Circulation becomes the infrastructure of ambition, the foundation upon which complexity can build. Imagine the ancient seas teeming with these early circulatory pioneers. Some are small worms gliding along sediment, others are soft-bodied animals with segmented bodies undulating with muscular contractions. Each carries a fluid that is no longer passive, a fluid that is beginning to perform tasks far beyond simple diffusion. Oxygen delivery, nutrient transport, waste removal, and signaling, all of these functions are integrated into a single, moving system. Evolution has discovered a new way to conquer limitations, movement from within. By the time true hearts emerge in early vertebrates, the principles of circulation are already well established. Muscular pumping, coordinated contractions, directional flow, and compartmentalization of fluid are all in place. The first heart chambers, simple tubular structures, amplify the efficiency of circulation dramatically. Oxygen-carrying pigments, now more advanced than the crude molecules of earlier life, move through vessels rather than cavities, ensuring that every cell receives the resources it needs to survive and thrive. The invention of circulation, once a simple ripple in a worm-like body, has become a transformative engine for life itself. Every heartbeat you feel today traces back to this ancient innovation. What seems natural and inevitable, the thrum of your own heart, the rush of blood through your veins, was once a radical experiment in a tiny, segmented creature crawling across primordial oceans. The pulse that carries oxygen to your cells is the product of millions of years of trial and adaptation, the echo of the first muscular contractions that pushed fluid through a soft, simple body. Circulation did not just sustain life, it enabled it to dream bigger, to move faster, and ultimately, to carry the story of survival through countless generations. The emergence of oxygen-carrying pigments marked a quiet revolution in the history of life, a moment when chemistry and evolution converged to create something that would forever change the possibilities for complex organisms. Before these pigments existed, internal fluids could transport nutrients and waste, but oxygen was always a limiting factor. It moved slowly, diffused unevenly, and left cells starved in the inner layers of larger bodies. The solution came in the form of specialized molecules capable of binding oxygen, holding it securely, and releasing it where it was needed most. This innovation may seem simple today, but in its time, it was nothing short of transformative. Early oxygen-binding molecules were rudimentary. They were small, flexible proteins that clung to oxygen loosely, and they appeared in various forms across different lineages of life. Some contained iron, some copper, and others relied on more exotic elements. The color of these pigments reflected their chemistry, red, blue, green, even colorless in some cases, but their function was far more important than their appearance. They were molecular couriers, ferrying life's most essential resource from the environment into the deepest recesses of the body. Without them, no animal could achieve the metabolic rates necessary for sustained movement, complex behavior, or long-term survival. In primitive multicellular organisms, these pigments began floating freely within the internal fluids, bathing cells in a tenuous supply of oxygen. For a small, slow-moving creature, this was enough. But as evolution pushed life toward greater size and activity, these pigments became concentrated within cells, forming the first true blood cells. This change increased efficiency dramatically. Instead of oxygen drifting randomly, it could be carried in precise quantities, delivered to the tissues that demanded it most. The evolutionary advantage was enormous. Organisms with oxygen-carrying pigments could move farther, hunt more effectively, escape predators more reliably, and thrive in environments that would have been deadly to less equipped competitors. The earliest pigments were simple, but they were the ancestors of the molecules that dominate blood today. Hemoglobin, with its iron atoms, is a refined version of these ancient molecules, optimized over hundreds of millions of years to bind and release oxygen with astonishing precision. Hemocyanin, used by mollusks and some arthropods, relies on copper and produces blue blood, demonstrating an alternative evolutionary solution to the same challenge. Both systems reveal the same principle: oxygen is precious, difficult to transport, and essential for life's expansion. Evolution experimented, selected, discarded, and refined until the optimal strategies emerged. Beyond oxygen transport, these pigments influenced other aspects of life. Oxygen molecules are highly reactive, and without careful regulation, they can damage proteins, lipids, and DNA. The pigments help control this reactivity, acting as buffers that prevent oxidative stress while enabling high-energy metabolism. In essence, they are not just transporters, they are protectors. The very presence of these molecules allowed animals to develop faster muscles, more complex nervous systems, and higher intelligence. They made possible behaviors that would have been unimaginable in the slow, energy-limited world of early life. Consider the ancient seas, where these pigments first began to appear. Small, worm-like creatures undulated through shallow waters, their internal fluids now carrying a new capacity for survival. Predators prowled, currents shifted, and oxygen levels fluctuated unpredictably. Creatures with pigments were no longer at the mercy of chance. They could venture into new territories, exploit richer food sources, and survive environmental swings that would have killed others. Life had gained a new tool, a chemical advantage that would shape evolutionary trajectories for hundreds of millions of years. The pigments also paved the way for further innovations in circulation. Once oxygen could be carried efficiently, organisms could develop closed circulatory systems, hearts with multiple chambers, and specialized vessels. Internal fluids became more than passive carriers, they became dynamic networks capable of supporting high-energy lifestyles. Muscles, brains, and sensory organs could flourish because the oxygen supply was no longer a bottleneck. The chemical revolution of oxygen-binding pigments was inseparable from the anatomical revolution of circulation. Even the color of blood, which we often take for granted, is a reflection of these ancient adaptations. The brilliant red of human blood comes from iron-bound hemoglobin, a design optimized over hundreds of millions of years for efficiency and stability. Blue blood, green blood, or pale blood in other species are all alternative solutions, each with its own evolutionary logic. Every hue tells a story of survival, experimentation, and adaptation to the pressures of oxygen availability, environmental chemistry, and metabolic demand. Blood color is more than aesthetic, it is the visible legacy of life's ongoing negotiation with one of the planet's most challenging molecules. Today, when you feel your pulse, remember that it carries the echoes of this chemical revolution. Every heartbeat moves hemoglobin molecules that descend from these earliest pigments, molecules that once drifted freely in the fluids of tiny, worm-like creatures. The oxygen in your veins powers thought, emotion, and movement, but it also carries memory of a world that demanded adaptation, experimentation, and innovation. Without oxygen-binding pigments, the story of vertebrates, mammals, and humans would never have unfolded. Your blood is not just a substance, it is a chemical testament to the ancient struggle for life, a bridge between the primordial seas and the conscious world you inhabit today. The evolution of red blood cells represents one of nature's most elegant solutions to a fundamental problem: how to carry oxygen efficiently to every corner of a growing, complex body. Early multicellular organisms had pigments dissolved in their internal fluids, but this arrangement was inefficient. Free-floating molecules could bind only small amounts of oxygen, and their delivery was slow and unreliable. As bodies increased in size and complexity, diffusion alone could no longer suffice. Life needed a container, a specialized unit that could hold, protect, and transport oxygen molecules while moving swiftly through the circulatory system. From this need arose the first red blood cells, the tiny carriers that would one day define the essence of human blood. These early cells were simple, single-purpose entities. Unlike the free-floating pigments of earlier life, they compartmentalized hemoglobin, keeping it safe from reactions that could damage other cellular machinery. Each cell became a miniature vessel, a self-contained oxygen courier, capable of ferrying life-sustaining molecules precisely where they were needed. This separation allowed organisms to increase their metabolic rates dramatically. Tissues deep within the body could finally receive enough oxygen to support faster movement, larger brains, and more intricate organ systems. The red blood cell is, in a sense, a tiny miracle of engineering, evolved through millions of years of trial and error to optimize survival. Shape was as important as function. The iconic biconcave disc of modern human red blood cells did not appear overnight, but its evolution reflects the relentless refinement of efficiency. This shape increases surface area relative to volume, allowing oxygen to diffuse in and out more quickly. It also provides flexibility, enabling the cells to squeeze through capillaries narrower than their resting diameter. Every curve, every indentation, represents an evolutionary answer to the demands of complex circulation, a solution honed by time and necessity, invisible to the naked eye, yet essential to life as we know it. Red blood cells also solve a chemical problem. Oxygen is reactive and can damage sensitive biological molecules if left uncontrolled. By isolating hemoglobin within dedicated cells, organisms protect the rest of their tissues from oxidative stress. This compartmentalization is a subtle but profound innovation, allowing organisms to harness the power of oxygen without succumbing to its dangers. It is a delicate balance, too little oxygen and metabolism slows, too much, and cells are destroyed. Red blood cells walk this tightrope with precision, a silent testament to evolutionary ingenuity. The evolution of these cells also set the stage for specialization in the circulatory system. Once oxygen could be transported efficiently, organisms could evolve closed circulatory systems with dedicated vessels and hearts, further enhancing delivery speed and precision. Red blood cells became the engine of energy distribution, enabling more complex organs, faster reflexes, and sophisticated behaviors. The ability to move oxygen reliably transformed life from slow, sluggish forms into agile, active creatures capable of exploration, hunting, and defense. Red blood cells carry another hidden legacy, the story of our ancestors' struggles with disease and survival. They are the product not only of biochemical optimization but also of evolutionary pressures imposed by parasites, pathogens, and environmental challenges. Variations in red blood cells, differences in shape, size, and hemoglobin type, reflect adaptations to malaria, altitude, and climate over millennia. The very structure of your blood is a record of battles fought long before humans existed, the imprint of selective pressures written in microscopic form. Consider the environment in which these cells first evolved. Shallow oceans, tidal pools, and early rivers teemed with life, predators, and shifting oxygen levels. Creatures with efficient oxygen transport could venture farther, escape threats more effectively, and exploit resources unavailable to their less capable peers. Red blood cells were not simply vessels, they were enablers, opening new ecological niches and allowing life to expand in scale, complexity, and diversity. The story of red blood cells is also deeply philosophical. These tiny structures, invisible to casual observation, carry the essence of survival, adaptation, and continuity. They are proof that life evolves not randomly but through countless small, incremental improvements, each one a response to specific challenges. Every drop of blood flowing through your veins is a living record of this ancient problem-solving process, a bridge linking you to organisms that existed hundreds of millions of years ago. When you see your blood, you see not just a fluid but a narrative of persistence, innovation, and the quiet triumphs of microscopic ancestors. In humans, red blood cells have reached a level of specialization that is astonishing in its precision. They carry oxygen efficiently, navigate a vast network of vessels, and contribute to immune function, pH balance, and even signaling within the body. Yet their origin remains humble, tiny, simple carriers that first evolved in soft-bodied, worm-like ancestors crawling in primordial seas. From these modest beginnings arose the circulating cells that make complex life, and conscious thought, possible. Every heartbeat, every pulse, every moment of activity is powered by these ancient innovations, flowing silently beneath the skin, linking the present to a deep, unbroken history of survival and adaptation. The heart, in its earliest form, was not the muscular, four-chambered organ we recognize today. It began as a simple tube, a rhythmic contractile structure that pushed fluid through primitive circulatory channels. For small, segmented creatures, this tubular heart was enough to move nutrients, oxygen, and waste efficiently. There were no valves yet, no precise directionality, and no chambers, but the simple act of rhythmic contraction introduced the principle that would guide millions of generations: the circulation of life is most powerful when driven by a pump. This innovation transformed survival from a passive struggle to an active conquest, giving organisms the ability to inhabit new ecological niches and push beyond the limitations of diffusion. Consider the early oceans where these tubular hearts first appeared. Shallow waters teemed with life, and competition was fierce. Predators and prey engaged in constant evolutionary arms races, and those with the ability to move fluid internally had a distinct advantage. They could support more active muscles, sustain faster metabolism, and explore territories their less well-equipped peers could never reach. The rhythmic contraction of these primitive hearts created a steady internal flow, connecting every cell to the vital resources it required. Life, once constrained by size and environmental fluctuations, began to expand in both scale and complexity. The tubular heart was not just a mechanical innovation, it was a solution to a chemical challenge. Oxygen and nutrients could now be delivered to cells at a much faster rate, allowing for higher energy output. Early oxygen-binding pigments, once floating freely in internal fluids, were now guided more efficiently to the tissues that needed them. Cells that had once struggled to survive in the inner layers of larger bodies could finally thrive. In this sense, the heart became a bridge between chemistry and anatomy, linking molecular innovations to the physical demands of multicellular life. Evolution experimented relentlessly with this design. Some early hearts remained simple, single tubes, while others began developing localized thickenings that would eventually become primitive chambers. Segmentation in the heart allowed fluid to move in a more controlled, directional manner, increasing efficiency and supporting larger bodies. These early modifications were slow and incremental, yet they demonstrate how even small structural changes could yield massive survival advantages. Each refinement was a step toward the highly coordinated hearts found in vertebrates today. The emergence of a pump also influenced behavior. With a more reliable circulation system, creatures could afford to become more active, exploring new environments and engaging in more complex interactions. Muscles could contract more powerfully and for longer periods, enhancing mobility, predation, and escape from danger. Circulation and movement became intertwined, the development of one drove the expansion of the other. The heart was no longer merely an internal organ, it became an engine enabling life to conquer space, energy, and opportunity. Over time, the tubular heart evolved additional features. Valves appeared, preventing backflow and ensuring that fluid moved in a consistent, efficient direction. Muscular walls thickened, allowing stronger contractions. Coordination improved, turning irregular pulsations into rhythmic beats that could sustain an ever-increasing demand for oxygen. Each of these innovations represented a response to environmental pressures, predation risks, and metabolic requirements. In essence, the heart became a finely tuned instrument, capable of delivering life with precision and reliability. This early evolution of the heart also laid the groundwork for closed circulatory systems, which would later dominate vertebrate life. Once a pump could efficiently move fluid through vessels rather than open cavities, the efficiency of oxygen and nutrient delivery increased dramatically. Organisms could grow larger, develop complex organs, and sustain high-energy lifestyles. The heart, in conjunction with red blood cells and oxygen-binding pigments, created a coordinated system capable of supporting activity levels that had been impossible for early, simpler life forms. Beyond biology, the evolution of the heart carries profound philosophical significance. It represents the first moment when life began actively controlling its own survival, rather than being entirely at the mercy of the environment. The pump transformed survival from passive endurance into active mastery, a subtle yet revolutionary assertion of agency at the microscopic level. Every heartbeat in modern humans echoes this ancient innovation, a rhythmic reminder of life's relentless drive to overcome limitation and expand possibility. From tubular contractions in ancient worm-like creatures to the four-chambered heart of mammals, the evolution of this organ traces a path of innovation, adaptation, and relentless refinement. It is a story of survival, but also of aspiration, life pushing against physical constraints, chemical limitations, and environmental hazards to achieve something greater. Every pulse of blood you feel today carries the memory of that first tubular heart, a simple yet transformative structure that set in motion the flow of life as we know it. The heart is not just an organ, it is the embodiment of evolution's capacity to invent, refine, and empower, connecting the smallest beginnings in primordial seas to the intricate, conscious existence of humans today. The evolution of a closed circulatory system represents one of the most transformative leaps in the history of life, a shift that allowed organisms to grow larger, move faster, and sustain higher levels of activity than ever before. Open systems, like those seen in many invertebrates today, are functional and versatile, but they rely on the free flow of fluid through body cavities, which limit speed, precision, and efficiency. As animals increased in size and complexity, the demands of oxygen delivery and waste removal could no longer be met by passive diffusion or open flow. Life required a more sophisticated solution, a network of vessels, a coordinated pump, and a fluid capable of transporting oxygen and nutrients efficiently. This solution would become the foundation of the closed circulatory system. In primitive chordates and early vertebrates, vessels began to form from previously open channels, enclosing the oxygen-carrying fluid and directing its flow. The formation of arteries, veins, and capillaries allowed for targeted delivery of oxygen and nutrients, ensuring that even the deepest tissues received a reliable supply. This structural innovation vastly increased efficiency, allowing organisms to maintain higher metabolic rates and support more active lifestyles. Muscles could contract more powerfully, organs could operate at greater capacity, and sensory systems could develop with unprecedented complexity. The closed system transformed the possibilities of life, enabling activity levels and behaviors that were previously impossible. With enclosed vessels, the importance of a powerful pump became more pronounced. The heart evolved to move fluid through this newly defined network, and rhythmic contractions became finely tuned to meet the demands of the body. Pressure could be controlled, flow could be directed, and oxygen delivery could be prioritized to critical tissues. This system also allowed for specialization. Certain vessels could focus on transporting nutrients, others on removing waste, and still others on delivering chemical signals. The closed circulatory system created a highly organized, integrated network, in which fluid, vessels, and pump functioned as a unified system rather than a collection of independent parts. The transition to a closed system also reshaped survival strategies. Animals with closed circulation could venture into new environments, escape predators more effectively, and sustain bursts of activity necessary for hunting or evasion. Energy-intensive behaviors became feasible because oxygen and nutrients could reach cells rapidly and reliably. Even the development of brains, complex sensory organs, and coordinated movement depended on this efficiency. Closed circulation made larger, more capable, and more adaptive animals possible, laying the foundation for the vertebrate dominance that would follow. This system also demanded refinements in the oxygen-carrying fluid itself. Hemoglobin-packed red blood cells became essential, as they could be carried through vessels with minimal loss and maximal efficiency. The combination of vessels, pump, and specialized cells created a feedback loop. Improved transport supported higher metabolism, which in turn selected for more sophisticated vessels and more efficient pumping mechanisms. Each component, heart, blood, and vessels, evolved in tandem, responding to the demands of survival and opportunity. The closed circulatory system also introduced a remarkable level of internal regulation. Animals could adjust blood flow to different tissues according to need, directing oxygen to muscles during exertion or to vital organs during stress. Capillary networks allowed fine control at the cellular level, ensuring that even the smallest, deepest tissues were nourished. This level of precision was impossible in open systems, marking a major evolutionary advantage. Animals with closed circulation could explore environments and adopt behaviors inaccessible to those relying on open fluid movement, expanding ecological niches and shaping the future of life on Earth. The system also changed the scale of life itself. Organisms could grow larger because cells were no longer limited by the slow, random diffusion of oxygen and nutrients. This size advantage translated into dominance. Larger animals could better compete for resources, escape predation, and exploit ecological opportunities. Evolution had created a structural and functional architecture capable of supporting life at scales and speeds previously unimaginable. Philosophically, the closed circulatory system represents a leap in life's capacity for control. For the first time, organisms actively directed vital resources internally, rather than passively relying on environmental proximity or chance diffusion. This internal command over survival echoes in the complexity of modern animals and even in humans, who rely on intricate circulatory networks to support consciousness, movement, and thought. Closed circulation is more than a biological system, it is a declaration of life's ability to manipulate its own conditions for survival. By the time vertebrates fully developed four-chambered hearts and highly branched vessels, the principles of closed circulation were already well established. Fluid, vessels, and pump worked in unison to deliver oxygen, nutrients, and chemical signals efficiently and reliably. This system made possible the active, adaptable, and intelligent creatures we see today, from the swiftest predators to the deepest divers. The closed circulatory system transformed life from constrained simplicity into dynamic complexity, enabling behaviors, structures, and capabilities that would have been impossible otherwise. It is a testament to evolution's capacity to solve problems elegantly, combining chemistry, anatomy, and function into a system capable of sustaining life on a scale that continues to amaze. The evolution of blood clotting represents one of the most remarkable and underappreciated innovations in the story of life, a mechanism so vital that without it, multicellular organisms would have been unable to survive even minor injuries. In the earliest life forms, damage to the body was often fatal.